So today we're going to be talking about B cell responses in the periphery. We've previously talked about T cell responses in the periphery. In fact, we just finished that up. And so we've been able to see the way that T cells can exert their effector function to get rid of pathogens, either as CD8 T cells becoming CTLs that can then kill infected target cells, or CD4 cells that can produce cytokines to affect some type of response. And now we're going to see kind of that same idea of what happens with B cell responses in the periphery and how our B cells are going to be able to perform their responses. So we left our B cells off when they finished their maturation in the bone marrow. And we need to now talk about what happens with that B cell that is IgM positive and IgD positive. It's finished development in the bone marrow. That potentially sees antigen in the periphery. We need to talk about what exactly is going to happen to that B cell in the periphery and look at its responses. Just as a little bit of a caveat, I want you to be aware that in reality, there are many types of mature B cells. Everything that I am going to tell you about has to do with what is listed here as a conventional B2 B cell. Sometimes it's also known as a follicular B2 B cell. There are other types of B cells, specifically B1 cells and marginal zone B cells, that vary in some aspects of their development and effector functions from what I am going to tell you about today. And I'm not going to go into a lot of detail about B1 cells or about marginal zone B cells. I just want you to know for your future life that technically when I told you about B cells and peripheral B cell responses, I was focusing only on one type of B cell responses, the B2 follicular B cell. In order to actually make a B cell response, we need to have a few different components in our secondary lymphoid organ. We need to have antigen in our secondary lymphoid organ for the B cell to bind and to activate that B cell. We need to have the B cell, because you can't activate a B cell if you don't have a B cell. So clearly you need a B cell. And we also are going to need T cells that can provide some T cell help. All of these components can be found in a secondary lymphoid organ like a lymph node. They will be found in unique areas. So in the top, you can see that there are T cell zones. There are follicles, which are really the B cell zones. And so there are going to be specific areas for the T cells and for the B cells. You can see on the bottom left that our T cells and B cells both might come in through an HEV and then travel to their respective locations based on chemokines. So you can see CXCL13 pulls the B cells to the B cell areas, while T cells are pulled to T cell areas based on other cytokines. You can also see that antigen, free antigen, is going to be coming in through the lymphatics. It's listed as, or it's shown as pink dots in this figure on the left. On the right, small and large antigens are distinguished, but in either case, those antigens are coming in through the lymphatics and are making their way into locations like the B cell zone, where you can see some of them here, or maybe carried in with a dendritic cell and can then make their way into the B cell zone. So we're going to need all of those components to make this process happen. For our B cell to become activated, we of course need signaling through our B cell receptor. We talked about the B cell receptor a bit in the past. We saw how the B cell receptor did not have a particularly long cytoplasmic domain, just like the T cell receptor, which will be on the exam on Friday. We saw how the B cell receptor gets around this by having two partner proteins, Ig alpha and Ig beta, also known as CD79A and CD79B, that have cytoplasmic domains containing ITAMs. Those are regions that can be phosphorylated so that we can start signaling. And when we saw some similar proteins in the T cell receptor, being CD3, that are really carrying out that same function in our T cells. And when we looked at activation of T cells, we also had a co-receptor protein that was involved in the activation of T cells. That co-receptor protein for T cells was either CD4 or CD8. And if you think about this process, what was the importance of CD4 or CD8 in allowing the T cell receptor to get turned on? Yeah, Nadia. Yeah, so CD4 and CD8 are closely associated with kinases. They're kinases from a family called the Sark family. Your book often talks about LCK, which is the one that goes with CD4. It's another related kinase that goes with CD8. The way that I presented the B cell co-receptor or the B cell receptor before, I sort of said, oh, there's also a kinase, and I kind of left out the details of that kinase. In reality, the reason why I left that out is that different types of proteins can do the job of being the co-receptor for the B cell. This figure shows one option, although there are a few different options of things that can happen. And so this figure shows a protein called CD21 acting as co-receptor, and CD21 would be interacting with a kinase in the intracellular domain, and you will see that better on the next slide. CD21 is a complement receptor. So if our antigen, like the antigens shown on this slide, has had complement bound to it, has had complement fixed to it, then we can have complement receptor binding along with our B cell receptor binding. That will bring a kinase nearby, and that will actually turn on our B cell. And so this is, in fact, I told you way back when we talked about complement, that complement fixation can turn on the adaptive immune system. And this is, in fact, one of the ways that can happen, is that if we have complement on our antigen, we can bring in a co-receptor that will have the kinase. But there are a few different potential proteins that can do the job here. This is the B cell signaling cascade. You can see our kinase. In this case, it's called Lin. Here, it's interacting with another potential B cell co-receptor called CD19. But I want you to think about this signal transduction model in big picture. If you look at the signal transduction model, do you notice anything about it? Does it seem in any way, shape, or form familiar to you? Yeah, Manny. We got NFAT. Anything else? What else? We got junk in June. We got RAS. We got DAG. We got PLC. It's basically the same. We've got a kinase. It's from the same family. It's a different one, but it's the same family of kinases. Lin is just like LCK. We've got phosphorylation of some other similar proteins. We've got a protein called SYK, which does the same thing that ZAP70 does. We've got some adapters like Blink that do the same thing as the adapters you saw. So it's actually basically the exact same pathway, which is why I figured I only had to really teach it to you once this year. It's not like I need to teach it to you more than once. But the same kind of themes are happening when we activate our B cell as the themes that we saw when we activate our T cell. If you think about activating our T cell, if you think about how we turn on a T cell in order to get really good T cell activation, we want to take our naive T cell and get excellent effector function. We want to get the best possible function out of that T cell. What do we need to do in activating that T cell? Yeah, Manny? Yeah, so we need multiple signals. This is sort of signal 1 for the T cell. We need to add signal 2 and potentially some cytokines as well. So this is signal 1 for the B cell. And you might ask, well, what about signal 2? Do B cells also have a signal 2? The answer is yes. But again, it's not quite as straightforward as with the T cell. With the T cell, there's sort of the one option. It's always CD28B7. With B cells, there are a couple different options that we can see for that signal too. And we can classify these into 2 different groups. One of them are referred to as T-dependent B cells or T-dependent responses. The other is referred to as a T-independent response, and there are 2 kinds of T-independent responses. I usually talk about this as T-cell dependent or T-cell independent. Your book is correct in that they were originally described as thymus dependent or thymus independent, but the thymus is where the T cells come from, so it's really the same thing. So first I want to talk about the T-cell independent activation of a B cell, which is shown on the right-hand side of this slide at the top. Our B cell can acquire a second signal through toll-like receptor or other pattern recognition receptor stimulation. And so you can see that happening in this cell labeled as B, where we have signal 1 coming through the B cell receptor and a second signal coming through a TLR. It could be some other pattern recognition receptors as well. There are situations where our B cell can get turned on. It's being shown here as sort of with one signal, but I would characterize it as this is generally an antigen that is polymeric. It's an antigen that is repetitive, and so we are likely going to have many B cell receptors being turned on. That's not depicted in this picture very well, but the idea here is it's a repetitive antigen. So B cell receptor is signal 1 and B cell receptor is signal 2 because you got so many B cell receptors here. Both of these are possible ways to turn on a B cell. You can see type 1 might be a cell wall component like LPS that's hitting a PRR. You can see type 2 is going to be a repetitive antigen. And both of these are good for turning on a B cell. However, the B cell that gets turned on in this way has some limitations. A B cell that is turned on in a T-independent response is not going to do class switching, it's not going to do affinity maturation, and it's not going to make a memory response. So you can turn on the B cell, but it's not a super good turning on of a B cell. I mean, it might be useful for you in terms of making some antibodies and not dying. In that way it's good. But it's not going to be kind of the full spectrum of all things that the B cell could do. Only if our B cell is turned on via an interaction with a T cell in a T-dependent response, which obviously depends on a T cell, will our B cell be turned on in such a way that it will be able to do class switch, that it will be able to do affinity maturation, and that it will be able to form a memory response. And so this sort of gives us the full activation of that B cell. In this case, the signal 2 is a little bit different than the signal 2 that we see in the T-independent cases. The signal 2 in this case is a protein on the B cell known as CD40, which binds to a protein on the T cell known as CD40 ligand. It's also CD154. So CD40 on the B cell can give that B cell its signal 2 so that we get full B cell activation and can get all of these types of B cell functions. this T cell is going to be making the CD40 ligand to hit the B cell and give the B cell CD40. And if you don't remember the details of CD40 and CD40 ligand, if you go back to YouTube and look at Shiv Pillai's Ode to Ben and Jen, he talks about the B cell showing CD40, or the T cell showing CD40 ligand to anybody. And so that was in there as well. Yep, Nadia. Can you tell me in which situation I say there's a lot of activation of B cells?
Like in T-independent, I think, yeah, when we were talking about like the polymeric repetitive antigens, having so many of them, is it that a lot more B cells get activated? Or?
I would probably say that it's a quantity thing, more so than a quality thing. And so here you can just see this shown in another way. The T-dependent B cell responses are going to give us our somatic hypermutation, are going to give us our class switch, and are going to give us differentiation into either a memory cell or another type of specialized cell called a plasma cell. So you can remember that with our CD8 T cells, for example, I told you that a CD8 T cell that comes out of the thymus is called a CD8 T cell. But once that cell has been activated and has gained its effector function, it's now a CTL. It has this ability to be cytotoxic. Similarly, our B cells can become memory cells. Our T cells can become memory cells too. We're going to deal with T cell memory a little bit later. Or our B cells can become a cell called a plasma cell. And I'll talk a little bit more about plasma cells. And this is all happening because of that CD40 ligand interaction between our helper T cell and our B cell. I told you when we talked about antibody responses that somatic hypermutation and class switch happen late in the response. They were not necessarily always part of the primary response. They were things that were part of the secondary response. And this is because before you can get your B cell activated, you have to get a T cell activated to help it. And so there has to be this delay in getting the T cells ready before you can get the B cells ready to make this response. So there are a few specific steps in making this response happen. The first step of the response involves the B cell interacting with antigen using its B cell receptor, or its antigen receptor. Our B cell is going to interact with antigen via its receptor, and there's going to be signal transduction that's going to occur, and that's not really shown here very well. That B cell is, however, going to become activated, which is shown here at the bottom. So our resting B cell is going to have some amount of MHC, some amount of B cell receptor, and once that B cell becomes activated, it will have more MHC on its surface. It will have other proteins that can help it stimulate other immune cells. And so you can see things that happen during B cell activation in this little box that came from figure 10-6. So our B cell is going to become activated as a result of signal 1 coming from this interaction with the antigen. But this part of 10-6 that I've cut out this B cell part from is actually a figure all about antigen presenting cells. And you need to remember that B cells are also one of our professional antigen presenting cells for class 2. And B cells have this ability to do receptor mediated endocytosis, which we talked about as being one of the ways we can acquire antigen for the MHC class 2 presentation pathway. And so not only is there going to be a signal transduction pathway that's not really drawn here, our B cell is also going to internalize the receptor along with bound antigen and degrade that antigen. That antigen will end up getting presented on MHC class 2 because our B cell is a professional antigen presenting cell. And that B cell will then present that antigen on MHC class 2. So this is sort of the first part of activating the B cell. Once the B cell has presented antigen on MHC class 2, you can sort of imagine it's now calling for a T cell and saying, hello, help me. If you see this antigen too, please help me. And so now the antigen that's being presented will be recognized by a helper T cell. Remember, this is MHC class 2, so this must be a CD4 positive T cell. And this will allow our T cell to provide B cell help using CD40 ligand. Specifically, this is a T follicular helper cell because we are in the follicle, and that we have recently seen as the job of the T follicular helper cell. As Manny correctly pointed out before, there often is a third signal using cytokines, which I'll say a little bit more about in a bit. And once the B cell has received this second signal as a result of T cell help, the B cell now can do things like become a memory cell, it can do its class switch, it can do its affinity maturation, and so on. This whole process happens in our secondary lymphoid organ, and both our B cells and our T cells are going to be moving around the secondary lymphoid organ quite a bit in order to make this process happen. So at the beginning, our T cells come in to the lymph node and they go to the T cell area. And our B cells come in to the secondary lymphoid organ and they go to the B cell area. Once the T cell is in the T cell area, the T cell needs to find a dendritic cell. It needs to see antigen from that dendritic cell and it needs to become activated. It needs to go from being a naive CD4 to being a helper T cell, particularly a T follicular helper. So if the T cell doesn't ever get activated, the B cell is just going to walk around sad, waiting to be activated. And we'll talk about this later, and we'll call it helplessness. And that is actually what happens to our B cells. Our B cell undergoes helplessness. Yes, Malik?
Then other immune cells can activate B cells, right? It's just dendritic cells are the best at it, or?
B cells need activation by a helper T cell. Dendritic cells are turning on T cells. Okay. So first our T cell has to get turned on. And then once our T cell gets turned on, our T cell moves to hang out near the B cell zone and be ready to help B cells. Once our B cell has seen antigen, it also moves basically to the junction of the T cell B cell zone so it can try to find a T cell. So our T cell has to see antigen from a dendritic cell, it has to get activated, then it's going to move to be by the B cells. The B cell is going to see antigen, present that antigen, and move to the T cell area so that ideally it can meet up with a T cell. And if our B cell is successful in finding a T cell, our B cell is going to move to a specific location in our secondary lymphoid organ. This location is called the germinal center. And it's a little bit tricky. I think sometimes people have trouble understanding this. I will admit to you that when I was in this lecture in my undergraduate immunology class, I was like, why is he talking in another language? Don't know what's going on. And really, it's not nearly as complicated as I thought it was. And so, because I was trying to figure out, well, where is this germinal center and what's going on in the germinal center and what's happening? A germinal center is the place that the B cells move to after they've been activated by a T cell, but a germinal center is also sort of the place where B cells are doing their reaction. And so in some ways, it's not like the B cell totally just moves to a germinal center that already exists. They move to a location and start doing stuff there, and that kind of makes it a germinal center. However, those locations are not just any old place. there is one really important feature of the location where the B cell moves and forms its germinal center. The B cell is going to look for a specific thing in that location in order to go to that location and make a germinal center. And that specific thing that our B cell is going to be looking for, ideally, if you just think about it, is that B cell is going to find some places where there's free antigen, where there might be antigen for its B cell receptor to bind. There is a specific place in our secondary lymphoid organ where all of the free antigen gets collected. So it's in a good spot for the B cells. This is on a specific cell type called a follicular dendritic cell. Good news about follicular dendritic cells. They live in the follicle. That's how they got the name follicular. Bad news about follicular dendritic cells. Follicular dendritic cells are not dendritic cells. They were named dendritic cells because they looked like them under a microscope when people first did histology of lymph nodes, but they're actually not a dendritic cell at all. Everyone loves that one. It's awesome. As a result, the key part of a follicular dendritic cell is not its ability to present MHC, specifically class 2. Don't think of follicular dendritic cells, no MHC. I mean, yeah, they've got class 1 because all cells have class 1. But ignore that. We don't care about their MHC. Follicular dendritic cells are these cells in the follicle, and our B cell is going to move to a follicular dendritic cell in order to form a germinal center. You can see a picture of a follicular dendritic cell here, where we have a cell body and all sorts of really long projections, and that's where it's got the name dendritic cell, even though it's not a dendritic cell. Follicular dendritic cells are important because they are covered with receptors. They are covered with receptors for complement. They are covered with FC receptors. They are covered with a number of different pattern recognition receptors. And the way I often think about follicular dendritic cells is that they're like a whole cell made out of flypaper. You guys know what flypaper is? You did not grow up in a very rural place like I did, where you definitely have to know what flypaper is. What's flypaper? Yeah, what is it? Huh? It's like a little piece of tape that you like hang from the ceiling almost, and the idea is that the flies fly into it and get stuck. And so follicular dendritic cells are just these cells that are super sticky. They have all sorts of kinds of receptors that allow any kind of antigen to bind to them. Because if that antigen has complement, it'll bind the complement receptor. If the antigen has antibodies on it, it'll bind an FC receptor. If the antigen has some mannose, it'll bind to a mannose receptor. So basically, this is just a cell that's sitting there having antigen stuck all over it. So it's a nice place for the B cell to try to find its antigen in order to get some signals. And so that's really what follicular dendritic cells are. And our B cell, once that B cell has acquired a signal from a T cell, is going to go to a follicular dendritic cell and start this process of a germinal center reaction. There will also, as you see in this process, be some T cells, specifically our T follicular helper cells, that are going to be at the germinal center as well. Germinal centers are very easy to see under a microscope if you look at sections of a lymph node or a spleen. And if you look at a germinal center, this is what it will look like. The germinal center has a couple of different areas. One of them is called the dark zone. The other one is called the light zone. You can see, especially on the right-hand side, a histology image where you can easily distinguish the dark zone and the light zone. Why do you think the dark zone is called the dark zone? Huh? It's dense. There are a lot of cells there. Light can't go through it very easily under the microscope, so it looks dark. But the idea with the dark zone is there are a lot of cells there. And why do you think the light zone is called the light zone? Yeah, there are fewer cells there. So we don't have and so the light can go through easier. It's light. You saw something similar to this with the thymus, with the cortex and the medulla. There was a lighter area and a darker area. And I told you there was something really important about that area that had lots of cells, sort of would correspond to the dark zone here. What was happening in that area of the thymus where there were lots and lots and lots of cells? Yeah, they were proliferating. They were dividing. I don't know why I put the emphasis on that in that way, but that's okay. We were getting cell proliferation there. We were getting cell division. And in fact, that's what's going on in the dark zone too, is that we're getting lots of proliferation. It's the same kind of concept. And so our B cell can become activated. You can see here's our B cell. It becomes activated as through T-dependent activation. And it can then go into the germinal center to perform something called the germinal center reaction. If that B cell gets a signal from antigen, it gets a little pat on the head. Your antigen is here. What is the first thing that cell wants to do once it gets its pat on the head? Yeah, Molly. Reproduce. We want to divide. We want to make more copies of that cell. That cell is useful. We need a lot of it. And so that cell is going to enter into the germinal center, and it's going to begin proliferating. You might realize that this probably is the dark zone, where this first proliferation burst is happening, because it's full of cells, and that's how it becomes dark. So our cell is going to go into this and start proliferating. But our cell is also going to do one additional thing in this part of the dark zone. Our cell is also going to turn on a process of mutation, where our cell is going to start mutating its heavy and light chain, listed here as SHM. Specifically, our cell is going to acquire a high frequency of mutations in the VJ region of the light chain and the VDJ of the heavy chain. And so you can see if we just plot mutation frequency over parts of the transcript or parts of the gene, there's a lot of mutations in the V and J and not a lot elsewhere. You can also see here the idea that the V region is going to pick up some point mutations that were not present before. And the idea here, the goal, the hope, is that our B cell is going to go from having a low affinity for antigen to having a high affinity for antigen. By acquiring some point mutations, we are going to have our B cell become better able to bind antigen. So the mutation process where we pick up all these mutations is called somatic hypermutation. When we do somatic hypermutation, the affinity improves, and we call that affinity maturation. And I will say that many immunologists, well, one of these is a genetic process, and one of these is a process that actually happens to a protein. Many immunologists use these terms interchangeably. So if we look at this, you can see how this might be a good thing for the B cell. But can you imagine a way that this could also be a bad thing for the B cell? Yeah, Emilio.
You mutate something that isn't the light chain, it could end up causing just bad stuff for the cell.
Okay, so if we mutate somewhere else, then bad things can happen to the cell. Absolutely. Yeah, Molly. So we could mutate and become self-reactive. Absolutely a possibility. What else? There's another big assumption that this slide is making. Yeah, Marina. Yeah, this is assuming we're going to have a mutation and get better at binding. We're going to get higher affinity binding. But in fact, it's just as likely that we could get these mutations and get worse binding. We could take our awesome antibody and ruin it throughout this process, just as easily as we could make a high affinity antibody. And so what do you think our B cell is going to need to do at this point, after it has done its round of mutation? What do you think, Manny? Check itself. It's going to need to test. It's going to need to test, does this still work? Does this still bind antigen? And so that is really what's going on in the germinal center reaction. Our B cell is going to do some somatic hypermutation, and then our B cell is going to try to interact with antigen on a follicular dendritic cell. And if the B cell interacts with the follicular dendritic cell and the cytokine, the B cell gets, and the helper T cell, the B cell gets some cytokines. It gets another pat on the head. And it says, you can divide some more, good job. And if the B cell can no longer interact with antigen on follicular dendritic cell and with helper T cells, then that cell is going to die by apoptosis. And so we can sort of see this process of evolving our B cells in the germinal center. So the way that this process will happen is our B cell will proliferate and undergo somatic hypermutation. One reason why the proliferation is incredibly important here is we're getting a whole bunch of copies of the B cell that's good. Let's imagine we get 1 million good B cells. Then we mutate all 1 million of those, and we assume we mutate them all in slightly different ways. And then we test all of those 1 million new B cells. Some of them are going to suck, and some of them are going to die. But some of them are going to be better. So all of those cells are going to interact with a helper T cell as well as with a follicular dendritic cell. I don't exactly know why it's shown all the way over there. But our cell is going to interact with antigen on an FDC. It's going to interact with a helper T cell. This is all going to happen in the light zone because we're not proliferating now. If the cell can make that interaction, it's going to get signal. If the cell cannot make that interaction, it's going to die. And a lot of this is going to be about competition. Competition for the antigen on the follicular dendritic cell, competition for the T cell. That's how we measure better binding. We make huge numbers of progeny B cells, and the ones that can outcompete their neighbors for binding to follicular dendritic cells and helper T cells must be the ones that have improved binding affinity, and that's how we keep going with this process. So if we happen to make, through our mutation process, some amazing B cell, it's going to outcompete the average ones. If we didn't make it, then the average ones are still going to win. And so when this process happens, our B cell, in fact, typically will go back to the dark zone, proliferate a bunch of times again, and try to mutate even some more. And we often will see multiple rounds of the B cell going through this germinal center reaction until the point where that B cell has made an incredibly high affinity antigen or an antibody that binds antigen well. Yep, Jordan.
So if they can go back and go through more, so
Yeah.
when do they die by apoptosis?
So they die by apoptosis when they can't get signal. So if they ever lose in the competition for FDCs and T cells, then they die.
They can have like low affinity and like go back. And
Yeah. So as long as they were able to be somewhat competitive in the first round, they can, go back. And if someone has the question of, well, when do they know they've done enough rounds, I do not know the answer. Yes.
So for the actual mutation, is it happening during the actual proliferation, or is it like, is there a mechanism after the proliferation?
It's happening right after the proliferation, but still in the dark zone. And there's a lot that it has taken us a long time to understand this mutation. So I'm going to show you some details molecularly of how the mutations work in a little bit. Okay. Also, if we look at the germinal center reaction, we see this affinity maturation happen, but we also see our cell differentiating to gain the ability to become a memory cell or to become a plasma cell. So that differentiation ability is gained while the cell is getting the signals in this reaction. And our cell undergoes class switch during this process. So our cell is going to, as you remember, our cell originally made IgM and IgD, and our cell is going to switch potentially to making one of the other antibody isotypes. And I just want to remind you that the constant region for IgM and the constant region for IgD are quite close to the VDJ region. And that's how we can get a transcript that contains both the IgM and the IgD constant region. And the constant regions for the other antibody isotypes are much further away, and we can't get a transcript long enough to contain them. But we can have this class switch process happen that's going to allow our cell to start to make those constant regions. Part of the way this works is based on which cytokines did our B cell experience when it was in the germinal center. So based on the types of T cells that have been activated and the types of cytokines that are being produced, so you can sort of imagine our Th1, Th2 differentiation happening here, we can get B cells making different types of antibodies. And so our B cell might switch to IgE if it receives a lot of IL-4 during its activation, while our B cell might switch to IgG2A if it receives a lot of interferon gamma during its activation. And you can imagine this happening because there are going to be T cells making a Th1 or a Th2 in that same lymph node while this whole process is happening. I will also say I am going to go with your textbook on this. I would have given you different cytokines for one of these isotypes, but I am going to agree with the textbook. So this is sort of a general part of how class switch works. And I'm going to say more about the detail of it eventually. When I talked about the constant region genes for all of our different antibody isotypes, I showed you that they were really far apart, but I didn't tell you about one important feature of that DNA. Right before each of the constant regions, there is something called this switch region, or it's labeled here as the S. It is completely not to scale. It is not nearly as big as the constant region. But you can see that before each constant region, there is a switch region, or a switch site. They don't show a switch site before D, but actually there is one. I don't know why they didn't put one there. And what happens here is that we will have another recombination process between switch regions. So we might have recombination between the switch region in front of C mu and recombination of the switch region in front of gamma 1 will delete the intervening DNA and suddenly put the gamma 1 constant region next to VDJ and throw away the intervening DNA. So class switch or isotype switching happens through another recombination process called class switch recombination, where we see recombination occurring at switch sites. There are a number of diseases in different patients where these processes are messed up. One of the most famous is known as hyper-IgM syndrome. In hyper-IgM, the patients have too much IgM. Surprise, surprise. There are two genes that are frequently mutated in hyper-IgM patients. So most hyper-IgM patients either have a mutation in CD40 or in CD40 ligand. So the B cells cannot acquire T cell help. Because the B cells can never acquire T cell help, they never are able to make germinal centers. They are never able to undergo the germinal center reaction. They can never do affinity maturation. And they can never make IgG or IgA or IgE, any of our other isotypes. So they have too much IgM because they can't have any of the other ones. These patients lymph nodes actually look very different than those of a normal patient because here you can see the germinal centers. They're really easy to see under a microscope. These patients also have no germinal centers because their B cells cannot get help from T cells. So we can see these types of patients. And there are two other things that can happen as a result of the germinal center reaction. So our cell also may be able to differentiate specifically into a type of cell known as a plasma cell or a type of cell known as a memory cell. And I want to say a couple of things about plasma cells. So plasma cells are sort of the differentiated type form of B cells, just like helper T cells are the differentiated form of CD4s, or CTLs are the differentiated form of CD8s. You can see the picture of a plasma cell here under the microscope. You will see that it has a nucleus, N, and you will see that it has rough ER. And basically, that's what it's got. This cell has become highly specialized to be a secretion machine. The cell has basically lost the ability to do anything other than secrete. Specifically, it's going to secrete high levels of antibodies. These guys are going to make boatloads of antibodies. All they're going to do is secrete antibodies. And so our B cell can become a plasma cell that is going to be basically an antibody factory that's going to be able to produce high levels of antibodies. That's the really good thing about plasma cells. Plasma cells don't have B cell receptors on their surface anymore. They don't have MHC on their surface anymore. They don't grow. They don't do hypermutation. They don't do isotype switch. They've already done those things. They do exactly one thing, which is make boatloads of antibody. Alternatively, our naive B cells have sort of the opposite phenotype. Our B cell, our plasma cell, because of this type of differentiation, is not really set up to live very long. It's not set up very well to live a long time for you. This cell is going to make you boatloads of antibodies for a short period of time to combat that antigen, and then it's going to die because it has gone to this dramatic specialization. However, some of the B cells that are coming out of the germinal center reaction instead are going to differentiate into cells that are called memory cells. These cells are specialized to be able to live an incredibly long time. We're going to have a whole day where we talk specifically about immune memory. But some of these cells are going to live for a long time to prevent you from getting, to help you fight that infection should it come around again. Yeah, Molly.
The plasma cells, do they replicate? Do they have the ability to
No.
replicate by themselves?
Nope.
Yeah.
They basically have one job, which is make antibodies, and they can't do anything else. So if this were my undergrad immunology class in 2000, I would be like, ta-da! B cell responses. And what you will notice in my ta-da B cell responses is that I didn't tell you how somatic hypermutation or how affinity maturation worked. I just said, magic, they're a thing. And in fact, that is the way that I was originally taught them, is like, and then these magical things happen, hooray. Since that time, we have learned the molecular processes that underlie somatic hypermutation and class switch. And so I'm going to tell you a little bit about somatic hypermutation and class switch. They both involve the same enzyme. And so in fact, a mutation in this enzyme also causes hyper-IgM syndrome. So it's the other big cause of hyper-IgM. If you don't have this enzyme, you can't do somatic hypermutation and you can't class switch. And before I tell you about this enzyme, I want to remind you of one important fact, which Chemistry Cat is going to remind us of. Chemistry Cat says that if I had a choice between DNA and RNA, I would choose RNA because it has U in it. You need to remember uracil. And you need to remember that uracil is found only in RNA, not in DNA. If there is uracil in DNA, something is wrong. An enzyme was discovered known as activation-induced cytidine deaminase. It's listed right there on top of that arrow. If you look at the word activation-induced cytidine deaminase, you might come up with an abbreviation that you would like to use for activation-induced cytidine deaminase. What abbreviation would you like to use here? Yeah, Molly? AICD. Turns out AICD was already used in immunology. When we talked about contraction, the contraction phase, contraction is sometimes called activation-induced cell death, AICD. So AICD was already used when this enzyme got discovered. So it's not called AICD. It's called AID. So AID is this enzyme activation-induced cytidine deaminase. Good news, it's only in B cells that are activated, activation-induced. And it has a very specific job. Activation-induced cytidine deaminase works on cytidine. You can see cytidine shown on the left here. Just for our refreshment purposes, what's cytidine again? Anybody? What's cytidine? What letter does cytidine start with? C. Where might you care about C? What, Jordan? Huh? Yeah, base pairs. This is the C, base pair. Is the C base pair in DNA? Is the C base pair in RNA? A similar version of it. So what happens with activation-induced cytidine deaminase is it reacts with the C's in DNA and changes this group, this amine group, to a carboxyl, which turns this from a C to a uridine. What's uridine? U. It's the U. So it changes C's in DNA to U's. Is there supposed to be a U in DNA? No. So this reaction alters the DNA. And then something has to happen to this DNA. And this is how both affinity maturation and class switch work. Both of them happen because of cytidine deaminations that AID is performing. I'm showing you figure 11-9 in two different places here. I've kind of split it up in two different ways. Because two different things can happen with AID. Sometimes with AID, we can get our uracil put into the DNA. And sometimes we can replicate across that with nothing happening. When that occurs, we'll get two daughter strands of the DNA, or two daughter DNA helices. One of them, it'll have the GA, which it had at the beginning, and it'll have the GA at the end, and so it'll go back to being right. It'll go back to being CT. The other one will become what you see here. Instead of having CT, we'll get a UT, and when polymerase tries to replicate that, it'll read that U as a T, and so we'll get a base pair change. We'll get basically a point mutation as a result of our C being turned into a U. We can also have situations where DNA repair enzymes look at this UG and say, no, 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 no. That's not supposed to be there. And actually remove that base through base excision repair. And other bases can be added, also giving us a point mutation. And so you can see single or basically double point mutations happening here, depending on the way that we repair that uracil. And so, in fact, somatic hypermutation is really what's happening because of the repair that happens to this uracil that we've added. And so in this situation, we started with CT and we ended up with CT and we had no mutation. But in either of these situations at the bottom, we've gotten point mutations in our DNA, which are these point mutations. They are mutations that are occurring in the V and the J region to allow for changes in specificity and hopefully affinity maturation. And then we just test the resultant B cell in order to make this process, make sure this process happens. One thing that you might guess from this, and this was known even before AID was discovered, was that there was a kind of predisposition to the fact that these spots that got mutated were pretty much always Cs. So they always started as Cs and became something else. People knew that for a long time. And that is really what happens. We can also see that pretty much all of these point mutations are targeted at the CDR regions, CDR 1, 2, and 3. And so you can imagine that this AID enzyme is targeted specifically to the CDR 1, 2, and 3 regions of heavy and light chain. So this enzyme is only going to be able to access that. It's not going to be able to access other genes, as Emilio mentioned, to potentially cause problems in those other genes. This enzyme is very well regulated. And what you might look at, and you might look at this and say, like, okay, I get how this cytidine deaminase can lead to somatic hypermutation. Like, we change a C to a U, we get a point mutation. Okay, I get how we get point mutations. This makes sense. But then for a really long time, people were confused about, well, how the heck can this same enzyme also give us class switch? Because in fact, the same enzyme leads to class switch recombination. And what we have realized is that it's simply a question of a different way of repairing the uracil. So we can also see situations where AID can lead to mutations, and the repair process can lead to a double-strand break. And you can especially think about situations where you have multiple mutations really close together, where there's a double-strand break. These mutations, where there are a lot of Cs very close together, are going to allow for class switching because they happen in the switch sites. So AID also targets cytosines at the switch site and hits them with a lot of mutations. But it's not just one base pair that usually gets hit in a switch site. That switch site gets hit multiple times with multiple cytosines at the same place. In that situation, we have double-strand breaks, and we have pretty severe DNA damage. In your cell, when you have very severe DNA damage in that way, the best way to perform DNA repair is by recombination, is through something called recombinational repair. And so, in fact, we have double-strand breaks made in the switch site, still the result of AID's action, and then the cell repairs all those double-strand breaks by recombining these switch sites. So recombinational repair happens when you have recombination between two really similar sequences, where the cell is trying to fix the sequence by using a similar one for recombination. The switch sequences are all really similar sequences, and so we get recombination between them as a repair mechanism, but it also gives us this deletion of the intervening constant regions. Yep, Molly.
So this is going to answer more guys, but is there any intention behind which switch regions get activated? Like, can you?
So Molly wants to know, how does AID know which switch region to target? If we want to do class switch to IgE, how does AID know it should be damaging this switch region so that we get recombination there? That's what Molly wants to know. And that is actually understood, or somewhat understood. So let's think about what this DNA actually looks like. This is a super long piece of DNA. Here's all the lengths of number of kilobases in between all of these sections. So if you think about that piece of DNA, is that piece of DNA sitting around like this in a cell, or like this, linearly in a cell? Got lots of head shake nose. How does that cell actually sit? Or how does that DNA actually sit in the cell? Yeah, Amelia. So it's bound up in chromatin. What kind of specific things might be happening? Is that a thing, Marina? Yeah, it's going to be wrapped around the histones. When the DNA is wrapped around the histones, those switch regions aren't accessible to AID. AID can't mutate them. The only way that AID is able to mutate a switch region is if the cell opens that part of the chromatin. And so the B cell is going to get a signal about which part of the chromatin to open. Should it open the chromatin by the IgE switch site? Should it open the chromatin by the IgG switch site? The signal to tell the cell which chromatin to open is from that cytokine. The cytokine that the cell gets induces a signal that opens the right part of the chromatin, the right switch site, so that we can get the proper type of class switch. Does that make sense? I know that answer very much involves molecular genetics, which everyone hasn't taken, but it is the answer to that question. Yes?
So do they know if it's like histones being shifted, or if it's...
I haven't kept up with that literature enough to be certain of the answer of that. They may know, but I do not. This is where we call my sister who works on B cells. Yes?
Is the break always happening at the later ones, or would it sometimes happen like M and D and then they would...
So, couple things for that. So it's actually happening at both. So both the M, the mu switch site, and say in this case the gamma 1 switch site are getting mutated, and you can see that when the recombination occurs, the final resultant switch site has parts of each of them. So you can see in the figure that this resultant switch site has part of mu and part of gamma. They were both mutated at the beginning. So your other question was about what goes on here. There is actually a switch site in front of D, and B cells can switch to be IgD producers and not IgM producers. And the biology of those cells is not well understood. As I've told you, the biology of IgD is not well understood, but there have actually been a couple of papers within the past month or so talking about what IgD producers and IgG switch B cells are doing, although it's not particularly clear.